Very good morning to everyone. Actually, we have been receiving the information on the waste decomposer developed by the NCOF Ghaziabad, and we wanted to develop a scientifically validate this product by studying in the different wastes which are having the different CN ratios. So with this aim, we have collected this culture from the NCOF, and we multiplied it in our ICR Indian Institute of Farming System Research Institute farm, and we are in the process of evaluating the different residues decomposition rate, both at lab and also pot culture experiments, and also in the field level. All the three studies we are undertaking, and also through network project on organic farming, we have identified the three centers, one at Modipuram, another one at Narendrapur in Calcutta, and the third one at Coimbatore, for complete biochemical characterization as well as the molecular characterization of this waste decomposer and its performance. So Dr. Dutta, who is conducting the lab and pot culture experiment, he will be explaining about the result of this particular product.
Actually, we have conducted experiments by taking the three different types of crop residue, that is rice, wheat crop residue and sugarcane crop residue, and we have applied this decomposer in the same dose in a different crop residue media, and here we are clearly identified that this is 1 month experiment, and there is a clear-cut difference between this control and in the decomposed treated samples. This is the control samples where the decomposition have not yet started, but in case of treatment, we have clearly mentioned, we have clearly indicated that there is already decomposition started, and it will mineralize the carbons present in the different types of crop residues. And this is the wheat crop residue where we have treated the decomposer in the systematic way for generating the scientific data. In case of control, there clearly it is indicating that there is no decomposition, but in the treated samples, the decomposition have already started, and this is the 1 month experiment, and there is a clear-cut difference that decomposition is actively started, and in the same result we are getting in case of sugarcane crop residue also, where the crop residue in sugarcane crop residue, this is the control, where the crop residue remain as such. Wheat, rice and sugarcane residue. So the pattern of the carbon mineralization will be changed.
Certainly. It depends upon the
substrate.
Yes.
Lignin concentration.
Yes.
So we have
so 30 days if you are actually it is noted that it started decomposition. And turning the color.
Turning the color.
How you are judging it?
Because we have the control. We have this one, the control. So there is a change.
There is change. Yes. This is the same color, brown color.
Yeah.
It is yes.
So there is a clear-cut difference between this control and
yes. So the 30 days start. So what I am claiming it is start decomposing.
Start decomposing.
30 days. So this farmers are seeing in the field.
Yeah.
Once they apply it 30 days, it is more visible this color.
Yeah. So you can easily compare this one.
This is the most decomposed.
Because these lines we have the
So quantity you have given the same or different?
No, same.
Same quantity. How much pure culture you have used?
No, pure culture. You have prepared
that culture you have applied the water.
And same as in case of sweet and in case of sugar.
Wheat, it is there. This is a undecomposed. It is controlled.
Yeah.
Close up. It's a close up.
And in one treatment, we have added the consortia with water. And there is consortia with equine.
ये वाटर क्या रखा हुआ आपने?
Sodium hydroxide. Sodium hydroxide.
Okay.
For starting the carbon trapping.
Okay.
For trapping the carbon dioxide.
Good. how much carbon it
Carbon
is
releasing.
releasing. Yes, that is.
And by this way we can estimate the activity.
That's true. So this is a wheat, then it is
This is
sugarcane.
sugar.
The sugarcane, what difference you are getting?
In case of sugar, this one.
This is a control.
Control. There is no decomposition.
Not this more decomposer. Yes. Then you estimate.
We will this what culture?
We have also contacted
Because
the.
indication alert started 30
Yes,
days.
yes, yes.
40 days we are claiming. So you see the 40 days.
So that's why you are.
No complete documentation. we
No,
will-
no, no.
So you are preparing this.
It is very systematic. All we take a photograph, everything, make it up.
Yeah. That's
एक अलग से आपका वो हाँ that's one thing I want to know. You have taken 3 types of material.
Yes.
Why you are taking 3 types of material?
because there are 3 types of crop residue. You have three different CN ratio.
What is the ratio of difference in this?
That is in case of rice 80:1, 200:1, 120:1.
So what do you mean by CN ratio?
CN ratio, this is nitrogen and carbon ratio.
So if,
And which is very much related for the initiation of microbial decomposition of any crop residues.
so which one is hard material?
Which one?
Is hard material. Harder material.
Hardened material, this one sugarcane.
Sugarcane is
Yes.
harder material. It is not easy to decompose.
It is not easy to decompose.
Nice. You can stand. Definitely stand.
Okay, sir.
You can start. Treatment you can tell what you are doing.
We have taken the four treatments. That is the control treatment, and another is the consortia with the effluent, and then consortia with water. This is the fourth treatment. Because we are applied some amounts of effluent as a source of energy in the initial period for multiplication of the microorganisms. That was our hypothesis.
What you are going to do it here?
We are studying how the nutrient is released from the crop residues in certain in particular days interval.
So you have used the decomposer in solution.
Yes.
How much quantity you have used?
I have taken the 50 ml
Solutions.
solution
In each.
in each
So observation will be
5 days interval.
5 days.
We are taking 5 days interval. The different types of all the types of nutrients
Okay.
which are related with the crop residues.
Okay.
And in that cases, we are also estimating the different fractions of the organic carbons
Okay.
which are released during the decomposition of microorganisms in the soil.
But it is without crop, no?
Yeah, it is without crop.
Achha.
Because in the soil conditions, how the microorganisms is doing, or what is the activity of the microorganisms in the soils. Our main emphasis study the activity of the
Other soil.
microorganisms.
But it is actually farmers are reporting it is more active in the standing crop also.
Standing
So etc.
So you have
any changes that also.
So we have already conducted another experiment in the field conditions
Okay.
also. Because here we have to study under the control conditions how much nutrient is released, how much different fractions of carbon is released in the soil environments. That is our motto.
How many days it is overall?
Now 1 month, just
1 month.
completed our 1
So
month.
any sample you have tested?
Yeah, I have tested the all type NPK and then micronutrients, different fractions of carbons.
So any difference is coming?
And there is a difference.
Difference it is.
There is a difference.
Somebody
Definitely there is.
difference is there.
Yeah.
So in all micronutrient as well as
In case of not micronutrient, it's called major nutrients.
Okay.
Primary nutrients,
Okay.
there is a difference.
it is different.
There.
But
Whether
once we
significant-
compile all the data,
Right.
when
then
we
only
compile,
we can
we can say.
here, sir, our main aim is to whether the microbial culture consortia alone is sufficient, or do we need to add the farmer manure as a starter
Yes.
dose for this
Starter
nitrogen
dose.
supply.
That's
So
why
that
we
was
have
the aim. We have mixed the microbial consortia along with the farmer manure.
Actually, what observation we have in field, when crop is harvested, so there is already your roots and everything is lying inside. The in situ decomposing we are telling. So then you apply this culture, it decomposes all the roots and release the nutrition. So here, as there is no crop.
Yes, there is no crop. That's why you have added.
That's why you are adding.
Yes.
Otherwise, it is not required to add it.
Yeah.
The adding is not required. It
It
is
will
crop
be more
residue.
effective in case of conservation agriculture or zero tillage
Yes.
systems.
Yes, it will be system because it is all residual
Yeah.
can be decomposed.
Can be decomposed.
So
But
this is
in
one-
case we don't have taken any plant sample.
नहीं production because main thing कि practically in farmers manure somewhere it is not available
and they are going organic farming. So whether in crop residual is enough to supply the nutrient and get the crop, what farmers are claiming in 20 rupees they are getting the total harvest, yield is at par, so that also required. Soil point of view, you have to test the soil and see how much carbon is releasing or what is carbon, what is uptake.
That's why you have taken one treatment, that is the microorganism consortia with water.
Okay,
With water.
okay.
And another treatment is FIM with consortia.
Then certainly a FIM will give more effect.
That's
It will
right.
certainly. But that is also there कि how much it is releasing. If it
Yeah.
is releasing the
How much?
binded form of your nutrient,
Yeah.
that is also.
That's
Is there any difference in
Is there
releasing
releasing
pattern?
releasing pattern
Do we need to add farmer manure or not
or
required?
not?
This will.
Or not required?
So moisture you are maintaining here.
Yeah, I master we are maintaining.